So welcome to today's session. My name is Amanda and I work for Accounting for Nature, one of the most credible scientific ways to certify and measure environmental condition. We are joined today with Luke Noble from EnviroDNA, who will talk about our most recently accredited aquatic vertebrate method that uses the very cool EnviroDNA technology. And so this method is one of the most flexible methods that we have in the Accounting for Nature framework. It can measure the presence of aquatic vertebrate species across a range of different scales and across a range of different confidence levels. But I'm not going to take up more time today. I will hand over to Luke Noble, who will go through the details of what this method has to offer.
Thanks, Amanda. So I'll be running through the method, as Amanda said, that we, in conjunction with Accounting for Nature and their Scientific Advisory Committee, have put together. And you'll see, I guess, if you haven't already looked at the document, that implementation of the method is really simple. So I think a main aim of this webinar will be to answer any additional questions you might have about eDNA in general, or some of the technical aspects of it. And I'll also give a bit of background about our company, EnviroDNA, what we do, and some of our current R&D that we're doing to kind of extend the eDNA toolkit. So I'm going to assume you're a fairly mixed bunch, and I'll try and keep it fairly brief and accessible, I think, and leave plenty of time for a discussion. So EnviroDNA has its roots in a research collaborative of the Center for Environmental Stress and Adaptation Research, based across a few different universities here. And CESA itself has now become a private entity, our sister or our mother company, I guess, focused on integrated pest management, conservation, and biosecurity. And so we share offices with them here in Melbourne. And together, our scientific backgrounds are across population genetics, quantitative ecology, biochemistry, molecular biology, statistical genetics, and data science. And so we try to operate at the interface between basic and applied science, ultimately just trying to get better outcomes for nature through monitoring and smarter management. And increasingly, we're supported by a great team of project managers and communications people and business development people down here. And so obviously we, I guess, like many of you, are really excited by the existence of organizations like Accounting for Nature, which are helping to get the numbers we so desperately need at sufficient scale and within a really robust interpretable framework that are necessary for an appropriate valuation of nature. We share these goals, and that's really at the heart of why EnviroDNA was formed. And so we've course believe that this new technology that I'll describe in a bit has an important role to play in this transition. So we've set up purpose-built labs to help deal with particular challenges of eDNA that I'll go into, and of course the protocols and procedures that go along with them. We have 4 segregated labs that sort of contribute sequentially to processing and analyzing samples as they move through our workflow, and we're continually investing in R&D and automation and information management, which obviously brings benefits to quality assurance, but really to efficiencies. And so we've been able to lower our costs over the last 6 months, even as prices, as I'm sure you know, are going up everywhere across the board. Because we recognize that cost is still a limiting factor for really widespread adoption at the scale that is required. And of course, as scientists, that we recognize that spatial and temporal replication are really critical for scientific robustness. So we're going to be continuing to try and really push the boundaries of how quickly and how well we can get eDNA developed so that it can be deployed at scale. And part of that, as I said, involves automation. So a lot of our processing is hands-off now, and at least as minimizing hands-on time. And this really enables much greater confidence in results. And also, importantly, we can trace samples and biobank them indefinitely, because as we're fond of saying that the only thing that really can't be repeated is sampling. So a major part of our goals is to enable really big scale baseline data that we can revisit in the future as technologies improve, as things are obviously changing quite quickly out in the environment. So to step back a bit, what do we consider environmental DNA? It really can be cast quite broadly, I think, as any and all DNA in an environmental sample. And that means it's any kind of sample that can be easily obtained by a human or some kind of device without really going to any effort to sort of isolate things in a way that has been traditionally done. For small organisms, microbes, that'll of course include, entire organisms and assemblages, living and dead. And then for larger organisms, that's pretty much anything that is derived from an organism directly or has come into contact with that organism. And so, as we're showing here, appropriately, the field really has started in aquatic environments, in fisheries management for obvious reasons. It's an excellent sort of transport and capture medium, and it continues to be really widely used in freshwater and marine systems. But there's also great promise for moving into terrestrial environments too, which is still an active area and something that's really emerging. I'll touch on that at the very end. So why has eDNA become useful so quickly? And I guess it stems from three main factors, and that's really sort of the surprising observation that DNA is actually quite stable and much more widely stable than people have assumed, and across a wider range of environments, at least that is once it has been sort of sheared into little pieces. So there's still enormous variability, of course, that we're getting better at dealing with, but to a first approximation, DNA is everywhere. I actually read this morning the record for DNA sequencing from ancient DNA, obviously in permafrost, the most ideal conditions, has been pushed back, doubled to almost 2 million years, and showing that there was once a rich forest and mastodons across much of Greenland. But even in much harsher conditions, for instance, here on the left, in arid and hot Australian environments, you can extract DNA that goes back tens of thousands of years. Here's a nice study looking at herbivore middens and the kind of things that you can find with the help of sort of natural accumulators like that. Secondly, obviously, is cost and how do you actually access all of this information? And obviously, I guess as many of you will know that the cost required to actually access this information through DNA sequencing has plummeted fairly recently. So it's now possible to actually use this information such that the sequencing is not the major part of the cost. And then thirdly, scientists have been really putting labeled sequences in public databases now for many decades. And so this is often fairly tireless and careful work of taxonomists and field workers. And it's usually necessary still to assign labels to all this information. People want to know what are the species. It's not actually strictly necessary to assess condition. You can do that without knowing what is in your sample to some extent. But certainly for this method that I'm going to talk about, and for most of the work we do, people are interested in knowing what is there. And that is possible, increasingly possible, as databases improve and we get a better handle on the diversity of life, at least the stuff that is easily accessible to us. That means large animals for the most part. So put together, the major advantages of eDNA, I guess, are that the traditional activities of getting out into an environment and sampling something, whether that's with a device or whether that's seeing things, and then the activity, the knowledge required to classify that, have now become separate activities. And so the bottlenecks on human expertise and reliance on human sensors are really relieved, and this, of course, dramatically lowers the cost per detection. And of course, indirectly, targeting large organisms from their DNA is not invasive for them. You don't have to touch something to know that it is there. This indirectness presents other challenges. eDNA is not the same as holding something in your hands, which not everyone is comfortable with. Although there are, of course, biases with traditional methods that are often really not made fully explicit. But eDNA is different, and it requires some different treatments. And frankly, all of these issues that I'm listing here, reference databases, how you interpret DNA, and the other kinds of information that we currently don't get from eDNA commonly, things like population genetic information or condition of animals or their sex. All of these are areas that are improving through time, although there will always, of course, be some information eDNA can never provide. But it's certainly a rapid development. And so the quality of data that we're generating now is going to be automatically improved with time as methods and databases improve. So getting baseline data, I think, is kind of a no-brainer at this point. And our particular journey to eDNA started mostly with work on platypus, before EnviroDNA was a separate entity. And so we still have a resident expert, Josh Griffiths, who is quite glad that we no longer have to do all-nighters setting fight nets. And the work that they've done showed that basically to get a 95% chance of detecting a platypus, you can either take 2 water samples of a given volume, or you can go out all night and set 6 to 10 fight nets. And I think there's no contest about which is easier there. And so we still do a lot of work in platypus, and we've gone from this kind of hard slog to at least statewide data and soon national data. Both, it's fairly dense now in Victoria, and it's becoming very dense in both time and space into New South Wales. And this particular scheme is one that we're working with Odinata Foundation and several state governments to roll out across Australia, so we can get a national map, obviously not just of platypus, which does not occur everywhere in Australia, but of all vertebrate species. Thereby generating, we hope, really useful baseline data for comparisons as we go on and uptake proceeds. And we're already seeing that that's extremely useful to detect change in time and to detect the impact of interventions or events like bushfires, floods, etc. So, lastly, finishing up on us, so we do the kind of work that underpins this method, broad metabar coding for various taxonomic groups, but we also do a lot of target group work where we will develop a specific assay, which is much more sensitive, obviously, than a general assay, where we're just looking for one thing in a complex mixture rather than trying to resolve a whole bunch of things. And so we have more than 70 assays for native and invasive species of particular interest. And then there's what we call metabar coding, which is this broad spectrum approach where we will try and amplify things that are conserved enough across a target group that we can actually amplify them in the lab, but they are different enough among species that we can then align them to a reference database and put labels on them at some resolution, usually species level for the organisms shown here. But for microbes, algae is actually quite challenging, diatoms, that kind of thing, then higher taxonomic levels. And these assays can be designed to be incredibly broad, for instance, across all eukaryotes, or more specific. For instance, we have a Decapod assay, let's say, if you're particularly interested in that group. And how that works is we go out, or clients go out, someone goes out, citizen scientists go out, and they will collect a sample from the environment, again, without really trying hard to make that sample informative. That can be soil, it can be water, it can be particular sort of general traps for insects, pitfall traps or sticky traps, if you're particularly interested in insects. It can be scats, which are obviously require a bit more expertise and yield sort of more restricted information on, say, diets or host genetics. And then increasingly, we're looking at, as I said, air for terrestrial monitoring, and that can be sort of rapid testing using unmanned aerial vehicles fitted with filters and fans, Or we think there's great promise in simpler, robust devices that can be left out in an environment for months at a time, essentially, for areas where the temporal resolution is not so important, but cost is and scale is. We're working on methods that will enable us to extract aerial DNA that is shed, say, from birds, from insects, from anything in the environment. And so we're currently working through those issues of what taxa are most amenable to detection, what are the effects of environmental condition. Air is obviously not water, and there are different challenges there with resolution. But we think there's enormous promise in it, and we have a bunch of projects at the moment and R&D where we have devices in fields generating data for us. So expect more in that area. But once we have our sample, then it's sent in to us. We bring it in, we extract DNA, we design or apply a specific assay to amplify a particular region of DNA in the genomes of our target organisms. So that is a amplification process that sort of amplifies trace DNA to usable amounts that is necessary then for high throughput sequencing, where basically we just read what is in this amplified mixture many times, and then align it to a reference database to resolve taxa. And the databases, as I said, are always improving. We make this a sort of core part of our activities to improve databases. There's many other fantastic public initiatives to directly address the database gaps, both in Australia and internationally. And so we will often try and source for a particular area any target taxa that are of interest and are not currently represented in a database and add them to that database. Sampling itself is really beautifully simple. It can go from these citizen science kits that are simple syringes and filters with preservatives that will keep things fairly stable, to dedicated equipment if people are doing a lot of this or doing it regularly, backpacks with water pumps and sort of other kind of hydrological information. It's really is quite simple. I've done it with my 8-year-old nephew. Terrestrial sampling, as I said, we are employing various kinds of pumps and traps, and that can be things that use the power of water to aggregate insect DNA, say, from leaf rolling or spray aggregation, or it can be these other kind of really large-scale aggregators that suck what is incredibly rare DNA out of the air and preserve it on site. All right, and lastly, obviously to make the most of this data, it's not just enough to go out there. You actually have to decide where to go and how often to go and all these kind of things. eDNA is a quantitative sort of probabilistic science. And so it's really important to have a good design of a monitoring program that will meet aims. And that means what taxa and what kind of change are you aiming to detect? Is this just a snapshot, or is it a monitoring program? And so this is the kind of thing that we engage with Accounting for Nature in developing this method. And for that, we borrowed data heavily. It's not the ideal data, but it's the best data that exists as far as we know. We borrowed data from a Melbourne catchment here, where eDNA has basically replaced traditional monitoring of aquatic vertebrates across the catchment here. And so in this program, we have good data over multiple seasons, multiple years, and for a lot of taxa. So we can use appropriate distributions of species and their frequencies to sort of parameterize a model for a much simpler problem in the case of Accounting for Nature, which is detecting aquatic vertebrate richness. This particular program has been designed to track quantitative changes in each individual species, which is a much harder problem. But for the purposes of putting in numbers on environmental condition, it is much simpler and therefore desirable to use something simple like aquatic vertebrate richness, which is simply a count of the detected native species in a given area through time. So we are all familiar, we're here with Accounting for Nature, and the aim here is to use an absolute measure of condition. So not just change through time, where that is arguably a much easier problem, but an absolute measure of condition that can be compared to an ideal state and across different environments. So there are 7 main steps to this method. These are fairly common to many AfN methods, and that is to define the purpose and scope of the accounting area, to bring together the existing data that's necessary to start to design a program, to split up that area into sensible assessment units that are representative of the site. This is actually a really critical step for environmental DNA and for richness, because essentially you're considering a count of one organism as the same as 100, as we'll go into. So stratifying it by various habitat types or known data is very important. We describe the indicators, which in this case are detections of species from eDNA, and we define the reference benchmarks, which can be quite a process using sort of pre-colonial ideals, if possible, and that is reasonably possible for aquatic vertebrates in most areas, but also bringing in local experts and as much knowledge as you can bring to bear, essentially, to define what should be the ideal reference. We collect the data, and that can be done by the proponents in this case, or with us, or we can provide training as we've done in the past. And then from there, the calculation of the scores are really quite simple. We've worked hard to really, I think, simplify this procedure, but we can go into some of the assumptions and underpinnings, if you like, in discussion. So a little bit slower. This is a case study for the Burnett Mary Regional Group, and that was recently completed. And so in this particular case, step 1 might be simply just to identify the area, to define the assessment units and what assets you are interested in monitoring. So in this case, and in the case of many NRMs, I suppose there is already an existing management structure that has been, based on the appropriate environmental variables. And so that can be used as is, or it can be further stratified, depending on the confidence level that you're trying to achieve. So as Amanda mentioned, we can basically meet any confidence level. It's a matter of, of effort and cost, obviously. So the sampling intensity will play a large part in the power to detect change of a given magnitude. So once you've done that, it's time to bring in existing data. So as I mentioned, that can be existing data on what species are there, ideally their frequency and their distribution, although of course that data rarely exists at large scale. That it would be the ideal data to input into a model that we can apply for each particular area to say this is an appropriate level of sampling and this is where you should sample. But as I said, often that data does not exist. You might have a species list, which is great, but the frequencies of those species and their distributions also matters in the realized power that you will get from a given sampling design. So we've had to make some assumptions, but again, we think they're guided by the best available data. And things we can improve as we go, basically, I think, if that is shown to be necessary. So in the case of BMRG, and this is, I think, common good practice, you would bring in riparian vegetation habitat types, for instance, if that's available. You'll bring in known drainage basins and local knowledge of vertebrate diversity. And then, of course, a major constraint is simply where can you access water bodies safely and reliably and repeatably. That can sometimes involve negotiations with private owners, or you can simply limit it to publicly available crossings and reserves. The stratification can be done in a number of different ways, either again using existing structure or by basically combining multiple spatial layers of numbers of interest, vegetation, habitat types, soil types, drainage flow, hydrological information, etc. So that can be something that either proponents can do if they have the expertise, or that we can help out with. And as I mentioned, it's really important that that stratification is representative and that it's reasonably granular, because this will give you more power to detect changes in richness. And so, in fact, the guidelines show that we recommend a sort of a rule of thumb, a sampling intensity for different sizes of areas, and also different minimum stratification of those areas, because of this relationship between stratification, sampling intensity, and power to detect change. They're all sort of interwoven. And so, again, we would like to tailor this to local conditions where recommended, although it's not strictly necessary. So the indicators here are, again, quite simply native aquatic vertebrates. And this may not necessarily be a point in which we all agree, but by default, we consider all native species of equal value. This is the number that we've sort of reached with accounting for nature, but obviously the data that is generated here can be used in many, many different ways. It can be used to monitor individual species. It can be used to look at native and invasive species or other kind of metrics. But for the purpose of simplicity and transparency, we've gone with richness for native species. Sampling, we provide consumables to do this, although it's not necessary that you use them, but it's quite simple. For the most part, if you're doing it large scale, you will want to use pumps, and these filters that we stock from Smith Fruit, for which we are the provider in the Australia-New Zealand region, are fantastic in that they are self-preserving. So you can do your sampling, wrap up your sample in a bag, and it will desiccate spontaneously and be stable there for weeks to months. And so they really don't need any special handling at all. The only requirement is that you get to places with a pump and you have the patience to sit there and sample. The filters are sent to us for analysis. And as I went through, that means we extract DNA, we sequence them, and we return to you a list, basically a matrix of sites by taxa, from which we or others can, other approved experts can calculate the richness indicator condition scores, which is simply the percentage of expected species for a given assessment unit that was actually detected in this sample. And then from there, we calculate the econ, which is a weighted average of the condition scores across the assessment units to reach this one number of condition. According to the method, you only have to do this every 5 years, but we would strongly recommend, I realize this is conflated with business interests, but from a statistical scientific viewpoint, it's really preferable, I think, to do this annually, or certainly more than from 5 years. Given that years like the ones we've just experienced are probably not necessarily very representative of normal conditions, whatever they may be. The Australian environment is really variable, and I think it's really preferable at this point, as we're still feeling our way with this method, to sample annually. And as I said, the sampling itself is easy. The costs, I think, are really reasonable for the quality and the quantity of the data that you get. So it's something we would strongly encourage. I'm happy to talk about costs if anyone wants some hard numbers in the discussion. Oop. And so then it's simply a matter of certifying your account and repeating. And so, as I've mentioned, the data really will improve with time. As databases improve, as methods that use other factors. I mean, BMIG is a great case study where it wasn't just aquatic native vertebrates that were measured, it was native vegetation and many other things. And so the integration of that kind of data will improve with time, so that you can be sort of backcast your data using the most available reference databases and analytical methods to make sure that your metrics are comparable, but they will be more accurate. So where do we see the future of eDNA going? It's moving pretty fast, but as I mentioned, we're already headed towards comprehensive national baseline data for at least for aquatic vertebrates and any vertebrates that are at least transiently associated with water. That does not cover everything, we understand. But we would also hope to get out of this data information for other taxa, not just vertebrates, because the temporal information is really important for detecting change through time, obviously. We can integrate other layers of information, as BMIG have done, looking at things like water quality, vegetation, etc., to improve these metrics. These are things that can be readily integrated with eDNA. And obviously, as we accumulate data through time, we will be able to provide better models of how these things interact to both shape eDNA signals and how they shape the biota that we want to help, essentially. So we can use it to, in a before-after sort of comparison study to show the effects of riparian revegetation or changes in water quality flows. And finally, expanding access to environments where fresh water doesn't capture everything, or there is simply not enough fresh water around. And so we're engaged in terrestrial monitoring, as I mentioned, from airborne DNA, and also in testing in marine environments. This is quite common in many areas now, whether that's looking for environmental condition from a broad range of taxa, or looking for particular species of significance. For instance, sharks is one group that we're involved in at the moment from a human safety perspective. And so we are going to continue on that path, and I hope you'll see other methods for a terrestrial taxa on the Certified with Accounting for Nature soon. And so with that, I guess it's time for questions.
Thank you so much, Luke. We had two questions pop into the chat while you were speaking. The first one is from Lillian, who's wondering how long does it take, how long does the process generally take for library preparation of samples for Metabar coding?
I'll answer the question literally, and then maybe if you mean how long until you get your data, that's a slightly different question, of course. But the library preparation itself can be completed for, let's say, 1 unit of samples, a laboratory plate of 96 samples within a day. And in fact, you can do a few more of those. That's reasonably quick. There are obviously many steps before that and after that, but the library prep process itself can be partially automated, and it can be done in a day.
Thank you, Luke. We had one from Anna wondering, when you use the pump, what kind of volumes are you putting through the filters?
Yes, this is important, and it also varies obviously across environments. We were generally aiming for 1 litre at least, but anything in the range of hundreds of mils up to several litres is good. I think the record that BMIG set was something like 40 litres in a abnormally pristine environment, which is a bit excessive and would have taken a long time. But yeah, around the litre mark is generally good, depending on your aims. I mean, the more important question, I think, is multiple samples. How do you spread them out, and how many do you need to take? But getting at least one good sample, a good volume for your samples is the first step.
Thank you. We have 3 questions from Dan. So the first one is asking if you can talk a little bit more about the temporal variability and seasonality, and how that might inform sample timing and also affect repeatability. Yeah,
it's a really important point. So, I mean, seasonal variation plays into it in a few different ways. Obviously, there's the biology of the species, which is going to play into any method. They differ in their behavior and how visible they are. So that is important to consider when you're designing your program, and that is why we would aim to sequence across seasons, ideally, and that's not always practical for everyone. And so from that sense, you're getting a slice through the environment. Basically, if you're sampling in one season, it may be the one that is most appropriate for your target taxa, recognizing that it will not be that appropriate or optimal for all. That is one part of it. The second part, obviously, is that as water flows change, you're getting dilution and transport effects. And so that is something that matters. There is simply not a data at the sort of the scale required to parameterize that into models. I mean, there are obvious things like, dilution matters and rate of flow matters. But when you're looking at really broad indices like vertebrate taxa diversity, you really can't put a model on every single species or even its species group to really make that data more accurate. And so that's where, as we do, I mean, the field is young, as we do more multi-year and multi-seasonal monitoring programs, we will learn how aberrant particular seasons are. So the short answer is that, yeah, we think it has an effect. We're pretty sure it has an effect. Certainly it has an effect on the volumes of water that you're able to sample if your conditions are challenging and turbid. But the signal in the DNA is less dynamic than, say, something like turbidity. You can still, we sampled with BMIG under really challenging conditions and still did a pretty good job, I think, of detecting a large number of taxa that were there. When it comes to localizing those taxa and the relationship between eDNA detections and organism abundance, then that's another question again. And so it's an area that the field needs to improve on, and that is only going to happen with implementation of more monitoring programs and collection of traditional data that can go alongside that to really sort of anchor these relationships for particular taxa. It's going to vary, for fishes versus mammals and birds, for instance. So that was kind of a long answer. Hope that hit it.
Thank you, Luke. Dan's last question was regarding if you have a standard price list available.
Yeah, we do. I don't have it. I can't bring it up on screen, but if you inquire, we can give it to you. And it varies a little bit with scale and, both the number of samples and the number of assays, that kind of thing. But as I said, the prices are going down, which we're really happy with. And so I guess I can give you a rule of thumb for this method. If you're at, say, property scale, you're looking in like the low thousands for a certification, and that's at minimum every 5 years. And then at the scale of something like BMIG, I believe that was around about 40,000 for a much larger program, including training, including supplementation of reference databases and all that kind of stuff. So I think, we think for the information you're getting, it's pretty cheap, and it's certainly much cheaper than you're going to get with any other method, I think.
Great, thanks, Luke. We have a question from Dane who's wondering, can you use eDNA sequencing to test for multiple organisms from one water sample collected, or is it one organism per sample?
Well, yeah, either is the question. If you want to optimize sensitivity, then we can do a qPCR test or a similar for one organism. But more commonly, and certainly for this method, we're analyzing a whole group. So that can be bacteria, all eukaryotes, or in this case, all aquatic vertebrates. So that is one assay for an entire group of organisms, or at least a sample of a sample.
Thanks. And excuse my ignorance, because I don't quite know what this is, but Henry was asking, what's the reliability of sampling for Ricali?
Good question. I have not been across any Ricali projects. I know we've had a fair bit of interest, and I would bet that our program manager, Josh, has an answer for this question. So I'm going to have to pass you along to him. But I mean, they are an aquatic mammal and they are intimately associated with water. I would bet that it is very good, as it is for platypus, for instance. It's kind of an ideal case.
I can jump in if you like, Luke.
Please.
Yeah, so I guess it's not reliability, it's more around sensitivity, and the sensitivity comes with the amount of DNA that's in the water samples, if we're looking at water samples. So Ricali are essentially a terrestrial species that jumps in the water occasionally to forage, as opposed to a platypus that lives in the water all the time. So we still do pick them up, but certainly the sensitivity for Ricali and other terrestrial species is much lower than those more aquatic species. We haven't actually quantified that, but I've certainly had I've certainly picked them up in plenty of places, but we do get negative results where I'm fairly confident they exist. So yeah, hopefully that, can't put a figure on it, but that's the sort of overall picture.
Great, thank you, Josh, for jumping in. Did you have anything to add to that, Luke? Are you happy?
No, I'm very happy to defer to Josh's expertise in that particular area.
Fantastic. Thank you, Josh.
I should just quickly say we are actually doing some modelling around detectability of terrestrial species from aquatic samples. So we'll be able to answer that question a bit better shortly, hopefully.
Easy. Thank you. The next question is from Benjamin, who's wondering, would species richness in an aquatic environment for this method be a reasonable indicator to inform overall water quality where other methods aren't available?
Well, we think yes. It is one of many methods that could be conceived of. We understand this, but we think yes, which is why we've used it in the method. I'm not sure if there's something else you were getting at there, but yes.
Fantastic, thank you. We have a question from Jennifer, who's wondering, how does the EnviroDNA Library integrate with others, such as the National Biodiversity DNA Library at CSIRO?
Yes, it's a great initiative that will be generating reference mitogenomes for all initially marine vertebrates, and then hopefully onwards to an entire atlas for Australia. And the answer is, so they don't talk yet. I mean, as far as I know, there's no publicly available data from that initiative yet. But we generate our own mitogenomes as part of assay developments, and we will certainly be looking to integrate with that kind of data. And I would assume sort of national scale detection databases that will be the next step, as you know, if and when that comes. We're in talks occasionally with ALA and people at CSIRO about this. So it's a discussion to be had in the future. At the moment, our database is, you know, obviously largely based on public resources that is available, you know, through GenBank, through other barcode curation activities like Midori and Bolds and various others, to which we apply, you know, our own curation and the addition of various data sequences that we get. I mean, most of the sequences that we get are shared with us by collaborators, and they end up in public databases anyway. We just maybe get them a little bit earlier because we need them now and we talk to people. So, I mean, most of our data is public, and we expect that it will integrate really well with these other activities. The other point to note, though, is that reference databases, even if you had a single instance of a reference sequence for every species, that's still not sometimes sufficient, because, you know, there is no one indicator cannot represent the entire species for a lot of taxa that are highly diverse or are structured into, you know, subspecies or anything like that. So there's always going to be room for additional efforts, even beyond the ones that are currently rolling out with CSIRO, to get more representative population genetic information for all species. So we think, yeah, between us, we will be contributing something to further the field for all of Australia.
Great, thanks, Luke. Next question is from Jess. Jess is wondering if there are any considerations or recommendations to reduce the risk of contamination of eDNA samples.
Yes, always an issue. And that may come from the person taking the sampling, or of course it may come from upstream, and does come from upstream. So we have guidelines certainly around best practice for taking water samples, and obviously handling them here in the lab. But also we can look at other ways, technical ways to reduce the influence of common contaminants like humans, let's say, or other farmyard animals in the lab, if we're interested in, you know, throwing them away and just looking at particular taxa. We can do that by over-sequencing, or we can actually do that by specifically blocking particular taxa from amplifying in the reaction. So yes, best practice for sampling is always recommended, and for handling, but even then, there is no guarantee that you're getting a clean sample. But it mostly involves fairly common sense things like wearing gloves, you know, and not getting into the water, and, you know, and the way that you actually just physically handle and package the samples. So yeah, we have guidelines on that. We provide training routinely to people to do their own sampling.
Excellent, thank you. Chris has a question. He jumped in a little bit late, and I was going to tell you, Chris, and everyone else who did, that the session is recorded and it will be uploaded. So in case you want to go back and watch it, the first few minutes you missed out on, you're more than welcome to do so. But to your question, Chris, how do you deal with high rates of false negatives and differing levels of detectability between species, not just based on abundance, but based on DNA shedding rates and behavior and movement? And how many samples per site or sites per catchment are needed?
Yeah, all good questions. So the way that this method, okay, there's a few different things there. I'll start backwards, I guess. The way that this method was designed was to provide assuming some constants out of necessity, to provide a certain sampling intensity for a certain assessment area structure that will provide a certain degree of confidence around a change of say 10% up, as set by the Accounting for Nature guidelines. And so to do that requires simulation with various parameters and detection probabilities. So as you would probably know, but this sounds like your question is quite well informed, the detection rates are not constant across organisms, and that information, as I said, simply does not exist for all of the organisms we want to measure. So yeah, it can't be done, frankly. We have to make some assumptions. We know detectability for platypus, we know detectability for some small native fish, for instance, and those detectabilities are generally quite high. So we can simulate across ranges of detectabilities, we can simulate across species distribution ranges, we can simulate across the way in which species may or may not differ in the way that they respond to an environmental change. All of these things can be, of course, simulated, subject to certain assumptions, and we'll get ranges and error distributions out of the models to inform us whether it's calibrated to detect a change. So we would love to be able to have the data to make the models better. But that, as I said, requires going and for different groups of organisms, actually testing that. So if we know a ricali is in the water in a given abundance at a given time, we would then go and sample, and we would then have a new number for ricali, and we would see, does that differ from platypus, from fish, for instance? And so we're very interested in continuing that work, and we are continuing that work with various groups to get that distribution of parameters that are important for the model. But we have to make, for these kind of methods, assumptions. And what we gain in scale and in sensitivity and the ability to get cryptic species that are beyond human sensorial constraints, we lose in some other areas. And we have to make assumptions to get that information. So it is not magic. There are new biases and new factors that affect eDNA, and our way forward is just to better handle them as better information comes along.
Thank you, Luke. That was the last question from the chat. If anyone else has a question, feel free to unmute yourself. I'm going to give you all a minute or so to do that before we end for today. Looks like we might have covered everything. If any of you joining today have any questions afterwards, feel free to send them either directly to EnviroDNA. You got the info email there. You can also send them to Accounting for Nature, and we will pass any method-related questions on to Luke. If you are jumping on here today and you don't really know what Accounting for Nature is, we do have a seminar series next week that you can also find on our Eventbrite page, which more so does a deep dive into the Accounting for Nature framework. With that being said, thank you so much, Luke, for joining us today. And if anyone wants to jump back on and watch the recording, that will be out on our YouTube channel next week.
Thanks, Amanda. Thanks, everyone, for coming.
Thank you.